Good day to you all. Myself, Dr. Shatrughan Midda, and this is a video recording. Okay, sorry. Let me just get this done. Okay. So, hello all. Myself, Dr. Shatrughan Midda, and I'm gonna teach you a little topic called ETC of subject biochemistry. So, let us just get started with the topic. Okay. So, ETC is electron transport chain. We transport an electron through a chain to produce our energy. It is the final step in the glucose metabolism. Glucose metabolism starts with when we eat food. Food comes into our stomach, it gets digested into simpler material like glucose, fructose, maltose, and the final product is glucose. It undergoes under the glycolysis and then through the Krebs cycle, and we got byproducts and subproducts around it. And the final thing is in aerobic metabolism is it goes through the ETC, that is electron chain transport or electron transport chain. Sorry, it is an electron chain reaction. Okay, which is it? See, electron in our body. Okay, let me just start writing few things, and we will get oriented with the concept here. We have already learned about what is electron. Electron is a negative charged particle, but in biology or in biochemistry we refer to as H. Okay, if H positive is proton, we know. Then plain H we refer to as electron in our textbook language here. See, whenever I need to mention some electron, if H plus is proton, then normal H is electron. That is the basic concept we need to move in. And chain, what do I mean by chain? There is a sequence of events occurring with this transfer of electron from one place to another inside the mitochondria or inside the cell where the energy is supposed to be produced. And reaction is supposed to be enzymatic reactions. See, any reaction in our body is caused by enzyme or it occurs spontaneously. These reactions are caused by a group of enzymes produced and located in chains to produce the final product of energy. So it is the enzymatic reactions. This much is clear to us. Electron chain reaction. Electrons are transferred in chain due to presence of by enzymatic reactions. It is electron transport chain. What is the site of the electron transport chain or ETC? Site in a cell, it occurs in mitochondria. We have learned that mitochondria is the powerhouse of cell, and now we will learn why the sad name is given to mitochondria. So mitochondria is a double walled. Mind my words, double walled. Sorry, I mistake. Double membrane, not walled. Double membrane structure / organelle of our cell. Why it is double membrane we will not get into it. We just need to remember कि ये double membrane है. One is outer mitochondrial membrane, one is inner mitochondrial membrane, and inside the inner mitochondrial membrane there is matrix. This is the basic structure of mitochondria. We need to get accustomed to it. Now, what is concept? Concept हमें क्या कहता है? Okay, if there is a charge particle, we have learned it in physics. If there is a charge particle and we make it move, if a charge particle is moving, it produces energy. Okay, it loses energy in form of heat, or it moves. If a charge particle is moving due to magnetic and other things, it produces energy. We are after this energy. We want this energy, and we want this energy to be stored. And what is the form of energy storage in our body? That is ATP. This is the basic principle. हम एक चार्ज पार्टिकल लेते हैं, ठीक है, उसको दौड़ाते हैं, उससे एनर्जी निकलती है, हम उस एनर्जी को स्टोर करते हैं और उसका एटीपी बना देते हैं. दिस इज बेसिक ETC. यही हमारा काम है. इसी का एक फैंसियर नाम आ जाता है, दैट अदर नेम इज बायोलॉजिकल ऑक्सीडेशन. We will understand it why it is called that. So we understand the basic concept. Now we need to understand कि ETC होता कैसे. इसके 5 parts थे. हमने कहा एक एंजाइम्स यूज होते हैं, एंजाइम्स इन चेन यूज होते हैं. तो कितने एंजाइम्स होते हैं? जितने भी एंजाइम्स यूज होते हैं हमने उनको 5 कॉम्प्लेक्सेस में बांट दिया कि ये 5 कॉम्प्लेक्स हैं. These are the 5 complexes in which these are divided. 5 complexes all are enzyme. What is a substrate? कोई भी एक एंजाइमेटिक रिएक्शन है उसमें 3 चीजें होती हैं. वन इज सबस्ट्रेट, वन इज द रिएक्शन एंड ऊपर एंजाइम एंड द प्रोडक्ट. प्रोडक्ट हमें पता है, हमें एनर्जी चाहिए. सबस्ट्रेट हमें नहीं पता, हमें एंजाइम पता है. वो कोई भी कॉम्प्लेक्स होगा उसको हम एंजाइम मान लेंगे उसका. व्हाट इज द सबस्ट्रेट फॉर ETC? सबस्ट्रेट इज NADH and other is FADH. NADH and FADH are substrate. 5 complexes enzymes act on it to produce ATP. This is the basic principle. Now if the substrate is NADH, then the sequence will be like first complex will act, then third will act, then fourth will act, then fifth will act. 1 3 4 5. If FADH is what the substrate is, then the sequence would be 2, the 3 4 5 is common, and that will produce us ATP. Two other things which we use are coenzyme Q and cytochrome C. These all are the parts or the basic parts of ETC कि हमें ये सारी चीज़ें चाहिए to produce ATP. Our final goal is to produce ATP. Our starting point is NADH and FADH. Okay. हम इन थोड़ा सा complexes के बारे में डिटेल जानते हैं. This is a diagram of mitochondria. Okay, I have made it a little bigger. We will discuss it. This is outer mitochondrial membrane. This will be inner mitochondrial membrane. I will label the complexes here in sequence. Okay, the complex 1, complex 3, complex 4. Similarly, complex 2, complex 3, complex 4. The same sequences 1 3 4 2 3 4. Okay, we are moving ahead. 1 3 4 2 3 4, and in between we need these. In between these complexes, I will label it as this is coenzyme Q, and this is cytochrome C. These are the parts. It is present. All these things are present in where? Inner mitochondrial membrane. And we will start our process. की होता कैसे है? होता ऐसे है कि just look closely. One NADH. NADH के पास क्या है? हाइड्रोजन. The complex one will do what? इसका जो हाइड्रोजन है ना यहाँ से लेता है and it will transfer it to coenzyme Q. Complex one will do what? It will take hydrogen from NADH, which is our substrate, and give it to coenzyme Q, which means coenzyme Q is been what? Reduced. Reduction and oxidation का basics ये होता है, जिसके पास हाइड्रोजन गया वो reduce हो गया. तो मैं कह सकता हूँ complex 1 ने क्या किया है? Complex 1 has done the basic thing that it has oxidized NADH, or I can say it has reduced coenzyme Q. So the name of complex 1 will become, it is NADH coenzyme Q reductase, because it is an enzyme which is doing the reaction of reduction. It is reducing coenzyme Q by transferring H into H from NADH to coenzyme Q. And while this reaction is occurring, there is a one phenomena occur for proton. What I am saying is for proton, जब ये reaction हो रही है, escapes from matrix and crosses the inner mitochondrial membrane and reaches the space between inner mitochondrial and outer mitochondrial membrane. So the proton gets outside into the space between both the mitochondrial membrane. The similar reaction will occur. Okay, now the coenzyme Q has got the hydrogen and it gets passed to cytochrome C with the help of which enzyme? Complex 3. So what would be the name of complex 3? It would be cytochrome C reductase, because complex 3 is doing what? It is reducing cytochrome C by taking hydrogen from the coenzyme Q and giving it to cytochrome C. Again 4 hydrogen will go and take benefit of this opportunity and leaks into the space between both the mitochondrial membrane. Then again the process occurs, and cytochrome C complex 4 would what do? Complex 4 will do, it will take the hydrogen away from the cytochrome C and give it into the matrix, where this hydrogen will act with oxygen to give H2O. Okay, these reactions occur in the matrix. पानी बना रहे हैं हमने कहीं से भी कुछ भी शुरू नहीं किया, हमने NaDH लिया, लास्ट में हमने मैट्रिक्स में पानी बना दिया, एंड द रिजल्टेंट व्हाट विल हैपन इज, इसके अंदर दो प्रोटॉन विल लीक आउट एंड 2H पॉजिटिव विल गेट इनटू द स्पेस बिटवीन द माइटोकॉन्ड्रियल मेम्ब्रेन, आउटर माइटोकॉन्ड्रियल मेम्ब्रेन एंड इनर माइटोकॉन्ड्रियल मेम्ब्रेन. अभी जो बाहर है ना, ये इकट्ठे हो गए 10 प्रोटॉन. Now what will happen is these 10 proton will go and find this channel, which is this channel. It is the complex number 5, which मैंने बताया नहीं. The thing is complex 5 is made up of two parts, F0 and F1. F0 is a proton channel and F1 is an enzyme. F1 is an enzyme. इस एंजाइम का नाम होता है ATP सिंथेस. तो यहाँ से प्रोटॉन आता है 4H पॉजिटिव व्हेन क्रॉस. आई ऑलरेडी टोल्ड यू, जब भी कोई चार्ज क्रॉस होगा उससे एनर्जी प्रोड्यूस होगी, एंड बाय वर्च्यू ऑफ दिस एंजाइम F1 ये जो एनर्जी है इसको कंजर्व कर लेगा बाय मेकिंग ATP. ATP बनेगा, ADP इसके पास होता है एंड इट विल प्रोड्यूस ATP विच इज अवर टारगेट. हमें यही तो चाहिए था, हमें ये चाहिए था और ATP हमारा बन गया. दैट इज हाउ द ATP इज प्रोड्यूस. अब इसके आगे क्या होता है, लेट्स से अगर ये NaDH ना होता, ये FADH होता, तो इस पे कॉम्प्लेक्स कौन सा एक्ट करता है, कॉम्प्लेक्स नंबर 2. कॉम्प्लेक्स नंबर 2 भी क्या करता है, 4H पॉजिटिव, यहाँ पे कोएंजाइम Q, यहाँ पे साइटोक्रोम C, H लेता है यहाँ से, H देता कोएंजाइम Q को, लेकिन इसमें क्या होता, 4H यहाँ से बाहर नहीं निकलता. दिस इज स्पेसिफिक थिंग. कॉम्प्लेक्स 2 कैन नॉट डू दिस. कॉम्प्लेक्स 2 हाइड्रोजनेशन तो कराता है, रिडक्शन तो कराता है कोएंजाइम Q रिडक्टेज, बट इट डजंट कॉज द ट्रांसफर ऑफ प्रोटॉन और लीकेज ऑफ प्रोटॉन फ्रॉम इनर माइटोकॉन्ड्रियल मेम्ब्रेन इनटू द स्पेस बिटवीन बोथ द मेम्ब्रेन्स. रेस्ट ऑल रिएक्शन अकर सेम. ओके, ये हाइड्रोजन नीचे देगा एंड यहाँ से 4H लीक हो जाएगा, यहाँ से 2H लीक हो जाएगा, अगेन विल सेम थिंग विल अकर हियर. So by virtue of FADH2 only 6H positive or 6 proton are getting inside the space between the two membrane. This is the basic principle. ऐसे ही काम होता है, इसमें कोई concept नहीं है. It's a story, we need to learn it in the sequence. I have made a little flow chart. Okay, maybe the above thing is not for you guys and you want a little basic stuff, then this is NaDH as a starting point. Complex 1 will be there. Complex 1 name would be NaDH, and it is giving two hydrogen to what? NaDH coenzyme Q reductase. Am I right? Reductase means addition of hydrogen. And by virtue of this, how many proton will get leaked? That is 4. Now next step is complex number 3. What will it do? It will take from the hydrogen from the coenzyme Q and give it to cytochrome C. So name of this enzyme would be cytochrome C reductase. It also add hydrogen, that is why the enzyme name is reductase. Again 4 hydrogen would leak into between spaces of this. Next sequence, complex 4, it will transfer hydrogen from cytochrome C into the matrix and make the water. And complex cytochrome C में से hydrogen is getting removed. If adding of hydrogen is called reduction, then removal of H will be called oxidation. Then the enzyme name will become oxidase. Complex 4 name is cytochrome C oxidase. And this is complex 5. I have told you F1, F0. There is NADH. NADH, how many proton will be generated? 10 H plus. And FADH 6 H plus. Now there is sequence if 4 hydrogen were passed through the channel. If 4 hydrogen, I said, pass through the channel, it will generate 1 ATP. So for NADH, 10 proton, it's a basic math. If 10 proton are there, it will generate 2.5 ATP. For FADH, 6 proton, it will generate 1.5 ATP per 1 FADH2. This is the bioenergetics of F1. F0 is proton channel. F1 is ATP synthase enzyme which converts ADP into ATP. These are all the things. Complex 2, if the product, if the substrate is FADH2, then the first name of this enzyme would be, one thing will be there is coenzyme Q and reductase. ये तो हमें पता है. It will be the coenzyme Q reductase. That would be same. What would be different from here? This was NADH. It would be succinate. Succinate, coenzyme Q, reductase. These are all the names of the enzymes of ETC. NADH, coenzyme Q, reductase, cytochrome C reductase, cytochrome C oxidase, and succinate, coenzyme Q reductase, and F1FO complex. These are the 5 enzymes. A little bit more detail or clinically oriented, if any of the process in this ETC gets hampered or stopped, what will happen? Patient will die because there will be no ATP. So anything which inhibits any part of this pathway will act like a poison to us, and we need to know which products are doing this. Specifically starting from the complex 1, there are few things like drugs and rotenone and pyridine. These will inhibit complex 1, that is NADH, coenzyme Q reductase. The next in our line is with the substances which inhibit complex 3. These are fenformin. There are many. Fenformin. Metformin can be also there, but fenformin, antimycin, and carboxin, and BAL, British anti-leucide. These are used in poisoning cases. Okay, so it inhibits complex 3. The inhibitors of complex 4 are ending with ide. Okay, these are azide, carbon monoxide, hydrogen sulfide, cyanide. Important. We are only discussing. There is a whole lot list. Okay, anything inhibiting succinate coenzyme Q reductase is malonate. Malonate is a substance which inhibits complex 2. Oligomycin. Oligomycin is a drug or substance which inhibits complex 5. And there is a one thing, ADP ATP translocase. Let's discuss this also. Here's this pink thing you can see in this diagram. What will happen is the ATP is produced, but ATP is inside the matrix now. We need ATP outside of the mitochondria and can be utilized in the cells. क्योंकि हमें cytoplasm में चाहिए, हमें nucleus के लिए चाहिए, हमें कहीं और भी चाहिए. So we need to transfer or translocate this ATP outside. So there is a tunneling system here. ADP will bind here, ATP will be released here. ADP will bind, it will turn, and ADP will come inside, and ADP will come inside and gets acquainted with the F1 site and becomes ATP, and this cycle will run. So this turning thing is enzymatic activity is called ADP ATP translocase, where ADP is getting inside and ATP is getting pushed outside. This can be inhibited by a substance called atractylosite. So ETC inhibitors are very important. We need to know them by heart. The concept is clear. There is one specific topic regarding ETC which is called uncouplers. What are uncouplers? These are basically if I want to generate, or if I want to give a specific term for this, is there are heat generators. Why are we calling them uncouplers? Because complex 5 does the reaction. The making of ATP or storing of energy is called coupling reaction. It's another name. Those who do not produce this reaction as a whole. What does uncouplers have? There is leakage. What I'm saying, there is leakage of proton. In the complex 5, where F0 is acting normal, see the channel is very much normal, the enzyme which is ATP synthase is defective. It does not work. So the hydrogen will pass and no energy will be stored. If the energy is not stored, and it will release. The most common form of energy release is heat energy. So uncouplers are, that is why, heat generators. We have got ourselves some natural heat generators and some synthetic heat generators. Natural heat generators are like a newborn. He does not shiver. The thermogenesis, heat generation, is a basic principle we use uncouplers. इनकी locations भी होती हैं. Nape of neck, adrenal gland, scalp, ये सब areas होते हैं जहाँ पे uncouplers होते हैं, present होते हैं. इनके examples क्या हैं? For the regard of biochemistry, we need to know the examples. The first example is thyroxine, thyroid hormone, thyroxine. The other is thermogenin, genesis of thermo, temperature. The next is long chain fatty acids. Long chain fatty acids are also acting as uncouplers. Next is unconjugated, not conjugated, unconjugated bilirubin. These are some basic examples of natural uncouplers. Now, one thing more I would like to discuss is one important synthetic uncoupler is 2,4-dinitrophenol. 2,4-dinitrophenol act as an uncoupler. Okay, these are the specific points which I wanted to discuss, and I hope you have a basic understanding of ETC and how the energy is being stored through it in the mitochondria. Why ATP is the powerhouse and mitochondria is the sole proprietor of energy of our cell. Thanks.